This is the first week of our online course. During this week, we will explain important basic principles of pharmacology and toxicology, how similar the two disciplines really are, but also what the differences between pharmacology and toxicology are. We will also describe a few of the major milestones that benefit us daily in terms of drug development over many hundreds of years. The learning objectives for this video are to become familiar with common terminology and definitions used within the disciplines of pharmacology and toxicology, to appreciate the similarities and differences between pharmacology and toxicology, to understand the major milestones that were accomplished in the fields of pharmacology and toxicology, and to become introduced to the important concept of the dose-response relationship. To start off, we will go over some definitions. Pharmacology is the science of drugs. This includes their discovery, what they do to living organisms, the specific chemical properties of a drug, how they work, and how they are used. Toxicology is the science of poisons and includes many of the same parameters as pharmacology. Based on these definitions, you can start to appreciate how closely related pharmacology and toxicology are. A drug is any biologically active substance, while a toxicant is a poison from any origin, and a toxin is a naturally occurring poison, except for metals. Some other terms you may hear or read about is a pharmakon, which means poison in classic Greek, and xenobiotic, which is a general term that is used for foreign substance taken into the body. It is derived from the Greek term xeno, which means foreigner. Xenobiotics may produce beneficial effects, such as a drug, or they may be toxic, such as a metal. Let's talk more about the similarities and differences of toxicology and pharmacology. These two subjects are really the same discipline at two different ends of the same spectrum. Pharmacology is the study of chemicals used at doses to achieve a therapeutic effect or response on an organism. When a chemical is used in this manner, it is a drug. Toxicology is the study of chemicals that produce a harmful effect on an organism. This means you could have a compound that at low doses is a great drug, but at high doses can cause toxicity, while other compounds may be toxic at any level. A chemical that can be a drug and a toxicant is described using a dose-response relationship. Paracelsus established the dose-response relationship with respect to toxic substances. His famous comment on this relationship is, "All substances are poisons. There's none which is not a poison. The right dose differentiates a poison and a remedy." He contended that experimentation is essential in examination of responses to chemicals. One should make a distinction between therapeutic and toxic properties of materials, and one can ascertain a degree of specificity of chemicals and their therapeutic or toxic effects. In pharmacology and toxicology, we need terms to help define how useful or toxic a chemical is and a way to quantitate results so different drugs and toxicants can be compared. A lethal dose is called an LD, and this is the lowest dose that causes death. The LD50 is the dose at which 50% of the animals tested die during some defined period of observation in an experimental study. This is useful to compare how harmful two drugs may be to humans. The effective dose 50 or ED50 is the dose of drug that produces the desired effect in 50% of a population. This may be an experimental setting or in the general population. The LD50 and the ED50 are a great starting point to understand how lethal and effective a drug may be, but to understand how safe a drug is, as in how close are the doses that could treat a disease and cause death, we need to determine the therapeutic index. The larger the TI, the more safe a chemical is relative to another with a smaller TI. The use of the ED50 and LD50 doses to derive the TI may be misleading as to safety depending on the slope of the dose-response curves for therapeutic and lethal effects. Some sciences closely related to pharmacology and toxicology include pharmacy, medicinal chemistry, and pharmaceutical chemistry. Pharmacy is the science concerning the preparation, storage, dispensing, and proper use of drug products. Medicinal chemistry is a branch of organic chemistry that specializes in the design and chemical synthesis of drugs. Pharmaceutical chemistry is also called pharmaceutics and is concerned with the formation and chemical properties of pharmaceutical products like tablets, liquid solutions and suspension, and aerosols. Pharmacology is divided into two main subdivisions, pharmacokinetics and pharmacodynamics. The relationship between these subdivisions is shown in this figure. Pharmacokinetics is concerned with the processes that determines the concentration of drug in body fluids and tissues over time, including drug absorption, distribution, biotransformation or metabolism, and excretion. A shorthand way of thinking about it is that pharmacokinetics is what the body does to the drug. Pharmacodynamics is the study of the actions of drugs on target organs. A shorthand way of thinking about it is that pharmacodynamics is what the drug does to the body. Pharmacology is one of the cornerstones of the drug discovery process. Originating in the 19th century, the discipline of pharmacology makes drug development possible. The medicinal chemist may create the candidate compound, but the pharmacologist is the one who tests it for physiologic activity. A promising compound is investigated by many other scientists, toxicologists, microbiologists, clinicians, but only after the pharmacologist has documented a potential therapeutic effect. This figure illustrates the development of major drug classes over the last 80 years. You're all aware of the serendipitous discovery of penicillin by the Scottish scientist and Nobel laureate Alexander Fleming in 1928. It was a fortuitous accident. In his laboratory in London, Fleming noticed that a petri dish containing Staphylococcus plate culture he mistakenly left open was contaminated by a blue-green mold. There was a halo of inhibited bacterial growth around the mold. Fleming concluded the mold released a substance that repressed the growth of bacteria. Now let's look at one of the major drugs to treat tuberculosis, isoniazid, which was discovered in 1945. Today, up to 2 million people die of tuberculosis each year. Despite the small but growing number of drugs that are effective at killing Mycobacterium tuberculosis, the current treatment is still burdened by the ever-increasing number of drug-resistant clinical isolates of Mycoplasma tuberculosis. Today, isoniazid is still one of the first-line drugs used for the treatment of TB. Before 1987, no antiretroviral drug was available, and treatment consisted of treating complications from the immunodeficiency. The first drug, Zidovudine, was approved in 1987. The figure drives home the limited lifespan of all drugs. Among antibiotics, penicillin started failing after about 10 years of use. The development of drug resistance is of great concern and has become a major difficulty in treating certain diseases successfully or with drugs that are affordable. Undeniably, much unfinished business remains in the area of drug discovery, drug development, mechanism of action, and mechanism of resistance. Throughout the ages, toxicological science has provided information that has shaped and guided society. In 1500 BC, poisonous plants and animals were recognized, including hemlock, opium, and the development of arrow poisons. In 1500 AD, Paracelsus developed the fundamental concepts of toxicology that all substances are poisonous at a certain dose. Orfila, a Spanish physician, is often referred to as the founder of toxicology. It was Orfila who first prepared a systematic correlation between the chemical and biological properties of poisons of the time. He demonstrated effects of poisons on specific organs by analyzing autopsy materials for poisons and their associated tissue damage. Toxicological science has advanced greatly with the discovery of two major methodologies. One was the principle of chromatography to separate and detect certain chemicals. Another major discovery in toxicology was the development of biomarkers to assess exposure to certain toxicants and to correlate those levels to clinical or other endpoints. This figure shows some of the milestones of toxicology. The file is available at toxipedia.org. Each entry is to provide a more detailed description of the individual toxicological event. Please make sure to look at some of these. Modern chemistry in the 19th century enabled scientists to synthesize new compounds and to modify naturally occurring drugs. Aspirin, barbiturates, and local anesthetics like procaine were among the first drugs to be synthesized in the laboratory. Semi-synthetic derivatives of naturally occurring compounds have led to new drugs with different properties, such as the morphine derivative oxycodone. Biologics represent the next exciting frontier of biopharmaceutical research. Biologics are a class of drugs based on proteins that have a therapeutic effect. These large protein molecules, which are composed of more than 1,300 amino acids and can be as heavy as 150 kilodalton, are essentially copies of optimized versions of endogenous human proteins. Biologics bind to specific cell receptors that are associated with a given disease process. Monoclonal antibodies are specialized biologics which recognize a very specific structure on the cell surface. There is a great deal of research going on in therapeutics for cancer and infectious diseases. Examples of biologics include monoclonal antibodies, vaccines, blood and blood products, gene therapies, and cell therapies. Last but not least, here is a very brief overview of the process of drug discovery, development, and approval. The development of a drug can be subdivided into 3 phases. The first phase is generally referred to as drug discovery and covers the search for a hit, its further development as a lead structure, and its optimization as a development candidate. The second phase covers preclinical development, the operations necessary to create the technical and regulatory requirements for the first application in humans. The clinical development phases that follow are comprised of more than 100 studies. The overall cost for single drug development is approximately $1.5 to $2 billion. And as you can see, the process can take up to 14 years for a drug to reach the market. Here is a figure from Nature Reviews Cancer demonstrating how integrated veterinary medicine, human medicine, pharmacology, and toxicology are in drug development before and after clinical trials. Through this integrated approach, it is likely that important questions about a new drug candidate can be answered before it enters human studies. For example, toxicity, dose regimen, pharmacokinetics, pharmacodynamics, and activity. Take some time to write out the answers to these questions and ask us any questions you have about this material. This video was a very brief overview, and you will be getting a lot more details in the weeks to come.